how do we use lethal photosensitization to treat infectious diseases? Yeah. So I'd rather you just wait until the end, unless it's a problem with what I'm saying or what. Yeah? Okay. Yeah, I'm quite happy to take any sort of questions at the end. So how can we use this process of lethal photosensitization to treat an infectious disease? The application of lethal photosensitization to treat an infectious disease is called photodynamic therapy, or PDT. And photodynamic therapy has been around quite a long time for the treatment of carcinomas. And so we're using photodynamic therapy for the treatment of infectious diseases. And it's interesting that although PDT of carcinomas started in the 50s and is now a well-established therapeutic modality, the first use of PDT was to treat Mycobacterium tuberculosis infections by a guy called Oscar Rabb in 1916, and he got the Nobel Prize for using PDT to treat infectious diseases. So we've come full circle here. So how would you use PDT to treat an infectious disease? Well, you would apply the photosensitizer, irradiate with light of appropriate wavelength, and this induces death of microbes, and also, it's something I'll be dwelling at length on later, inactivates virulence factors of bacteria. I personally think that, notice this phrase here, topical application of the photosensitizer. I think that PDT of infectious diseases is going to be by and large restricted to topical infections rather than systemic infections, because it's difficult to imagine how you could systemically administer one of these light-activated agents and then have the individual undergoing whole body irradiation, both internally and externally. Internally doesn't even bear thinking about it. It could be very uncomfortable. But so I think largely topical infections, and of course a wide range of topical infections, most of the oral infections, burn infections, wound infections, and the like. So there's quite a lot of potential there. So what are the ideal attributes of a photosensitizer? First attribute should be that it absorbs red light. Why red light? Well, it gets us away from the danger of using blue light and low wavelength light with the dangers of mutation, etc. But also red light penetrates tissues quite well, and a lot of topical infections are not just really on the surface of the body, they also may involve or compromise the outer layers of the epithelium. Red light will also penetrate mucus and will penetrate proteins. There's usually some organic moieties associated in secretions of individuals when these infections take place. There must be a rapid transition from the singlet to the triplet state, a long-lived triplet state, a high quantum yield of photoproducts, and obviously must be of low toxicity. And the photosensitizers we've investigated at UCL, we've looked at a wide range of photosensitizers. We've concentrated mainly on the phenothiaziniums, i.e. methylene blue and toluidine blue. We've also worked on cyanins, porphyrins, and chlorins, and phthalocyanins to some extent. These are the photosensitizers which are classically used for the PDT of carcinomas. But in our experience, these are not very useful for killing microbes. These are very, very potent antimicrobials. These not so good. We've looked at a wide range of organisms in our research. Obviously, we've done a lot of work on oral bacteria. That's just a partial list. We've looked at the classic periodontal pathogens, P. gingivalis, Aggregatobacter actinomycetum comitans, my favorite bug, Prevotella intermedia and Eubacterium species, a wide range of periodontitis and endodontal associated organisms. But also we've looked at extraoral infections as well, you know, things like Staph aureus, Strep pyogenes. We did a lot of work on Helicobacter pylori, and I still think that this could be a very useful, we even went as far as using animal models in, what was it again? Some strange animal, will come back to me, mongooses or something. Helicobacter pylori, which is really a surface infection of, or surface colonization of the gastric epithelium. So, and it's possible to apply a photosensitizer to the gastric epithelium and using an inflatable balloon distribute light throughout the sort of gastric epithelium. So we haven't done a lot of work. We used to do a lot of work on that, less work sort of recently. So there are a number of target organisms we've looked at. But what are the sort of factors which would influence the outcome of an interaction between light, a microbe, and a photosensitizer? Well, classically, the same kind of factors, the same kind of outcome variables that would apply to an antibiotic, except you've got the additional factor of light. So first of all, you have photobiological factors, the photosensitizer concentration, the light energy dose, light energy density, and the pre-irradiation time. In other words, how long do you have to apply a photosensitizer to a microbe before it becomes sensitized to killing? Then we have microbial factors. For example, the growth phase of the organism. This is very important indeed. We all know that antibiotics do not kill non-growing organisms. Most antibiotics only work on rapidly growing organisms in the logarithmic phase of growth. So the growth phase of the organism is very important. Whether the organism is in the planktonic state, in other words, free-floating in an aqueous environment, or whether it's in the form of a biofilm. Now, for oral infections, of course, this is particularly important because all the sort of significant oral infections are caused by bacterial biofilms. And whether the organism is in pure or mixed cultures, one can imagine that pure cultures might be more easily sort of killed than mixed cultures, because in a mixed culture you might get preferential uptake for one organism over another. And then we have environmental factors, the pH, the nature of the suspension fluid, for example, whether it's saliva, blood, or serum. So I'm not going to show you all the data we have. We've looked at all the major perio-pathogens. We've done all the studies on many organisms. So I just want to sort of concentrate on P. gingivalis just to give you an idea of the kind of data we get. Now, these experiments are extremely complex. We've got so many variables and we need so many controls. One has to consider the possible effect of light on the organism by itself. One has to consider the effect of the photosensitizer on the organism by itself, and then the combination of light and the photosensitizer. So lots of controls. For some reason, the error bars are missing from this. I do apologize. So this is a culture of Porphyromonas gingivalis, very high density, 10 to the 10 colony-forming units per mil. This is our control with no light and no sensitizer. This is our control where the bug has been irradiated with light in the absence of a sensitizer. These green bars are the bug which is kept in the dark, but irradiated, kept in the dark and exposed to the sensitizer. And then we have our test bars, these yellow ones, which are the bug irradiated with light in the presence of the sensitizer, and we're using different light doses. So as you can see, for all these controls, the light by itself doesn't affect the viability of the organism. The photosensitizer by itself doesn't affect the viability of the organism. But the combination of light with the photosensitizer, you get a dose-dependent decrease in the viability of the organism. So we're going from 10 to the 10 down to less than 10 to the 1. So we're getting a sort of 10-fold, 10 log reduction. This shows the effects of different pre-irradiation times. So in this case, what we did was we exposed the bug to the photosensitizer, in this case toluidine blue, and we're using toluidine blue in conjunction with a helium neon laser, because the peak absorbance of toluidine blue is at 633 nanometers, and the emission band of the helium neon laser is also 633. You must match your photosensitizer up with your light source. So here we treated the organisms with the toluidine blue for 300 seconds, 60 seconds, or 0 seconds before actually irradiating. And you can see that basically this is an instantaneous photosensitizing effect. So there's no need to incubate for long periods of time before lighting up. It's basically an instantaneous effect. This shows the effects of growth phase. There are 3 phases of growth in any organism. We have the stationary phase. So if you take some bacteria and you put them into a nutrient broth, Nothing happens for a while. It's a time of adjustment to the new environment, and this is called the stationary phase. The bacteria then, because of differential gene expression, start sort of growing quite rapidly, producing the right enzymes, the right metabolomes for coping with that environment, and grow rapidly. And then eventually they run out of one key nutrient and we get a stationary phase. Stationary phase cells lag phase cells are very, very resistant to antibiotics. In general, most antibiotics are only effective against rapidly growing cells. And you can see that in the case of PDT, that the three growth phases are more or less identically susceptible. So there's no real effect of growth phase. This is with using toluidine blue and a helium neon laser. This business of the generation of resistance survivors has always been an issue, although as I pointed out earlier, we can't really see what mechanism would be involved. So what we've done, this just shows two experiments, we've done this repeatedly over 30 generations. We've exposed an organism to a suboptimal light dose and suboptimal photosensitizer concentration, so we get a lot of survivors. Then we've taken those survivors and re-exposed them and got the survivors, and taken those survivors, re-exposed them, and done that over many, many generations. And we've never detected any resistant isolates, resistant strains at all. We're obviously interested in the mechanism of kill, so I won't go into this to some extent. These are TEMs, transmission electron micrographs, of Porphyrimus gingivalis before and after lethal photosensitization with toluidine blue and Heaney laser light. And this is our controls, and these are our organisms once they've been exposed to the light in the presence of the photosensitizer. And you can see that in the case of the killed organisms, you can see extensive vacuolization and cell wall alterations. So it's affecting the outer membrane and the cytoplasmic membrane and causing vacuolation of the cells. This is an SEM of P. gingivalis, and it's rather interesting. You can see that after lethal photosensitization, the cells appear to clump, and we think this is cross-linking of the outer proteins. This is a TEM of P. gingivalis before and after lethal photosensitization using tritiated toluidine blue. We wanted to find out where the photosensitizer actually went to. Now, although these look very, very similar, if you actually sort of count the number of particles, you can see that after photosensitization, there are far more internally than externally. So there's certainly sort of cytoplasmic membrane disruption and outer membrane disruption. If the mechanism, as we've proposed, is by singlet oxygen and or other radicals, then we should be able to get increased kills by carrying out the experiments in deuterium oxide. Singlet oxygen extends the lifetime of deuterium oxide extends the lifetime of singlet oxygen. So if singlet oxygen is involved, we should certainly get increased kill in deuterium oxide. And you can see these experiments here. This is P. gingivalis suspended in water and irradiated with 0.73 joules of light in the presence of toluidine blue. And here is the same conditions, but in deuterium oxide rather than water, and you can see an enhanced kill. When you increase the light dose even further, this is the suspension in water and the suspension in deuterium oxide. If you increase the light dose to 4.4 joules, you can see that in the presence of deuterium oxide, we get greatly enhanced kill. So this supports the idea that it's singlet oxygen mediated. This is an experiment looking at the membrane fluidity of P. gingivalis. P is a value, the reciprocal of which indicates the extent of the degree of membrane fluidity. And you can see that in the organisms that have been treated with laser light and the photosensitizer, the membrane fluidity has decreased remarkably. So, and once you decrease membrane fluidity, that has a knock-on effect. It interferes with transport systems, energy generation systems, and all sorts of other important metabolic activities. So it does interfere with membrane fluidity. We've also actually identified some of the photolabile proteins in P. gingivalis. This is a Western blot of P. gingivalis, and this is looking at a particular region of that before photosensitization and after photosensitization, and concentrating on these proteins, protein A, protein B, and protein C. And you see that these particular proteins are absent in the Western blot after lethal photosensitization. So they've disappeared, something's happened to them. And when we sort of did an N-terminal sequence analysis of these proteins, we found that this particular protein was identical with the Arg1 beta protease of P. gingivalis. And this is another section of the Arg1 beta protease. And protein C was identical to the lyse gingipane. So we've identified what were the particularly photolabile proteins in P. gingivalis: the arginine-specific thiol protease and the lysine-specific cysteine protease. So I haven't got time to go through all of the different mechanistic studies we've done, but to summarize them, we found that deuterium oxide enhances killing, suggesting that singular oxygen is involved in the process. We know that free radical scavengers inhibit killing, for example, tryptophan. If you carry out the experiments in tryptophan, you get far less killing. There's evidence of lipid and protein damage. We can detect in Western blots the disappearance as well as the dimerization, trimerization of certain proteins. And also using a malondialdehyde assay, we can ensure that there's lipid peroxidation. And this would account for the disruption of the cytoplasmic membrane. With evidence of membrane leakage, certain intracellular enzymes will leak out once you photosensitize and radiate these organisms. There's a decrease in membrane fluidity, and we have actually detected the sensitizer triplet state and singular oxygen. So to summarize a lot of, preliminary laboratory studies, we find that we can easily kill large numbers of organisms by low light doses. Low concentrations of the photosensitizers are effective. We can get kills at all growth phases, and short pure radiation times are effective. We've carried out the experiments in serum and blood. This does have an inhibitory effect, but not total inhibitory effect. So we get fewer kills. The kills are less effective in the presence of serum and blood, but you can overcome this by using higher concentrations of the photosensitizer and higher light doses. We've also carried out experiments on mixed cultures and found there seems to be no particular specificity. You can kill bacteria in mixed cultures, and the kills are generally unaffected by the pH. So all these results would suggest that PDT may be effective for the treatment of oral microbial diseases caused by P. gingivalis and similar organisms.